Here you go. Great, thanks a lot. I want to start with explaining where I work and what I do. So I'm the technical director of the Upstate Freshwater Institute here in Syracuse. We've been around since 1981, and we're a not-for-profit organization. So I get paid, but the company can't make money. There's about 20 of us, so it's a very small research institute. We conduct research on lakes, like Onondaga Lake, rivers, and streams. We have 6 boats that we take out to collect water samples and make measurements. And we have our own water chemistry laboratory where we analyze the samples that we collect. Our mission is to provide the scientific basis for the protection of fresh waters, lakes, rivers, and streams in New York State, and to advance education and research related to these fresh waters. The way we know if we're doing a good job is by how many articles we publish in journals. For scientists, that's very important. That tells people how credible your work is. When you do work, it's kind of graded by 3 other experts in the field. And if they say it's good enough, then they'll publish it in one of these journals. And we get about 10 articles a year published, which we're pretty proud of. We do research on a number of different lakes in New York State, including the Great Lakes, Finger Lakes. Today we're going to be talking about Onondaga Lake, which you guys are almost experts on by now. You probably know almost as much as I do. And Onondaga Lake is right here. Right now we're right in this area. So, in particular today, I'm going to talk to you about a project that we've been doing on the lake since really started in 2005 in terms of thinking about it, and the actual project started in 2011. And it has to do with adding a chemical called nitrate to the lake to prevent a certain form of mercury, in this case methyl mercury, from accumulating in the lake and getting into the fish. And listed here are the folks that worked on the project with me from the Upstate Freshwater Institute, from Parsons, and from Syracuse University. I know you've heard quite a bit about this today. I think you know that the sediments of the lake are contaminated with mercury, and a variety of other contaminants. We're going to focus only on mercury today. Mercury concentrations are elevated in the water, in the sediments, and in the living organisms of the lake. You've heard a lot about the remediation program. There are a lot of components to it. Today we're going to focus on just one small piece of this project, which is nitrate addition to the lower waters. So, I want to talk a little bit about mercury in aquatic ecosystems. That's just fancy language for lakes, rivers, and streams. How does mercury get into these water bodies? Well, primarily from electric utilities, incinerators, when they burn garbage, for example, and industrial manufacturing, which was the case for Onondaga Lake. We're particularly concerned about one form of mercury called methyl mercury, and it's abbreviated MEHG. And we're worried about that form because that's the form that bioaccumulates in living things. I think you heard a little bit about bioaccumulation earlier, if I'm not mistaken, but I'm going to explain it a little further in the next slide. But the problem with methylmercury is that it can affect nervous systems, your brain, spinal cord, other nerves in your body. And for us, for humans, and for wildlife, we get most of our mercury from eating fish. So in Onondaga Lake, we're particularly concerned about mercury accumulating in fish. This looks like kind of a complicated chart, but it really isn't. What these are levels of mercury in the water and phytoplankton, which are microscopic plants out in the lake. Zooplankton are microscopic animals that eat the phytoplankton. Planktivorous fish are little fish that eat the zooplankton. Piscivores are bigger fish that eat these fish, and loons are a bird that eat the fish. So pretty much this is a food chain going from just the water all the way up to the birds that eat the fish. And these are mercury levels in each of those organisms. This is kind of a funny scale. In science we call this a log scale. So instead of being 1, 2, 3, each one of these ticks is a factor of 10. So what this means is that in big fish and in loons, mercury concentrations are about 1 million times more than they are in water. And that's why we're concerned about this. Really low levels in water can result in really high levels in bigger organisms. All right, before we go much further, I want to talk a little bit about stratification in lakes. Relatively deep lakes, like Onondaga Lake, stratify during the summer. And stratify just means layered, right? So there's different layers, like there are layers in a cake. There's fancy words, epilimnine, which just means upper waters, metalimnine, which is the middle waters, and hypolimnine, which are the lower waters. And basically, during the summer, these upper waters are warm, and they have a lot of oxygen and nitrate. The lower waters are cold and dark, nothing lives down there, and the oxygen and nitrate levels are very low. And that becomes important because our work is really focused on this lower layer. This is where the mercury accumulates during the summer. Okay, this slide tells us a little bit about mercury cycling in lakes, how mercury moves through lakes. In general, methylmercury comes from the sediments at the bottom of the lake, and when there's no oxygen or nitrate at the bottom, it's released into the lake water. And it's really not a problem down here, but in fall, when the lake destratifies, this mercury gets up into the upper waters where organisms like fish can take it in. And that's really the problem. If the mercury always stayed in these sediments, it really wouldn't be a problem. It wouldn't get into the food chain and wouldn't get into the fish. The guys that produce methylmercury in the sediments are called sulfate-reducing bacteria. You've probably heard of bacteria, right? They can make you sick. Also, we need them. If we didn't have bacteria in us, we couldn't digest our food. Bacteria are everywhere, but there's a certain kind of bacteria called sulfate-reducing bacteria that convert mercury from a relatively harmless form into this toxic methylmercury form. And those guys live down in the mud. Okay, so the problem is that methylmercury accumulates in the lower waters of Onondaga Lake during summer. And that's shown here. This is the level of methylmercury, and these are the months of the year in 2009. And we can see that during summer, when we get to late August, September, into October, these values are really high. We've got 4s and 6s. Outside of that interval, we're down here, almost can't even measure it. So the problem is really this, and we want to stop that. the units that we measure it in are nanograms per liter. Probably never heard of that. I hadn't heard of it until not that long ago, but 1 nanogram per liter is really, really small. It equals 1 part per trillion. 1 in a trillion. It's equal to 1 drop in 20 huge Olympic-sized swimming pools. So it's a really, really small amount. So why do we care? Well, we care because of this bioaccumulation thing, right? That even though this is a tiny amount, by the time it gets up through phytoplankton and zooplankton and fish and birds and us, it can become a pretty big amount. So that's why we want to stop that accumulation. All right, well, like I mentioned earlier, if this methylmercury always stayed in those lower waters, it really wouldn't be a very big problem. The problem happens when the lake mixes in the fall. This methylmercury that's in the lower waters causes an increase in the upper waters. So this stuff really, there wasn't any fish coming in contact with it, not that big a deal. This is a big deal because that's how it gets into the fish. So, you guys seen this before? Does this look at all familiar? The scientific method? Okay, cool. So the first thing we want to do, we need to ask a question, right? And our question was, how can we stop methylmercury from accumulating in Onondaga Lake? We want to stop that spike in the lower waters so we can stop that spike that happens in fall. So that's the question we asked ourselves back in 2005. How are we going to do this? And what we noticed was that this spike in methylmercury only happens when this chemical called nitrate, it's a form of nitrogen, gets low. So we looked at the data year after year, and what we saw is when this gets really low, down below 1, close to 0, mercury gets really high. And so from that, we developed a hypothesis, which is the next step in the scientific method. And our hypothesis was, well, if we keep these nitrate levels high, the methylmercury won't come out of the sediments, and it won't be a problem. All right, well, now we have a hypothesis, so now the next thing we generally want to do is get some more data, see if this makes any sense. So we did a little exploration and some research. So back in 2005, we thought we had a pretty good idea. So folks from the Upstate Freshwater Institute and from Syracuse University approached Honeywell and approached the New York State Department of Environmental Conservation and said, you know, we think we've got a pretty good idea. Are you guys interested? They agreed. They said it's worth further research. But of course, they're not going to take our word for it that it's a good idea. They wanted us to do a lot of studies to prove to them that this was worth trying. So in 2006, we did a feasibility study. In 2007 and 2008, we did some other studies, including the sediment incubations. This is kind of a crazy picture here. You can make it out, but these containers down here contain sediment from Onondaga Lake. And we exposed that sediment to different concentrations of nitrate to see if the mercury came out or not. So it's kind of a complicated setup, but before we tried it in the whole lake, we wanted to just try it in these small containers to prove that it worked. And it did. So we did some more studies in 2009 and 2010, and then by 2011, we were actually ready to try it in the whole lake. And that was called a pilot test. So you're trying it at the full scale of the lake. So now we're ready to test the hypothesis. And the only way to do that for sure was to start adding nitrate to the lake. So we built a barge. And actually, I don't know if you guys saw, but the barge is parked down there as a Honeywell sign on it. But basically it looks like this. It's a big barge with 2 tanks of nitrate on it. This nitrate has to be diluted, so water's pumped in, it's mixed on board this barge, and then injected down into the bottom of the lake. We're not adding it to the top of the lake, we're adding it to this hypolimnian or lower, colder waters, right down near the bottom. So that's basically how it works. There's a picture of the barge out on the water. These are the 2 big tanks of nitrate. They contain, I think it's about 2,400 gallons apiece in that neighborhood. This is the pipe to get the water from the upper part of the lake to dilute the nitrate, and then the nitrate goes down through these pipes down to the bottom of the lake. So here's a map of the lake. We broke the lake up into 3 sections. Cell 1 down here, cell 2 in the middle, and cell 3 further to the north. And we figured out that if we added nitrate at each of these locations, each week, from the beginning of July through early October, that would be enough to keep the mercury in the sediments. That's what we hoped anyway. So each time we went out, we added 4,600 gallons of nitrate, typically on like a Monday, and then a Wednesday, and then a Friday. And then we go back out the next week and go Monday, Wednesday, Friday again. And did that all summer long. So once we added the nitrate, we needed to find out if it worked, right? If it did what we wanted it to do. In order to do that, we had to collect and analyze a lot of data. And in order to do that, we used this instrument, which is pictured here, that's called ISIS. And it's really cool because what it allows you to do is make measurements in the lake without bringing any samples back. We used to have to collect water, put it in a container, bring it all on the boat, drive it over to the lab, and then the folks in the lab would have to test each of these samples to tell us how much nitrate is actually in the sample. This instrument, we just lower through the water column from a cable. It just free falls, and it makes a measurement every quarter of a meter, which is about 10 inches. So every 10 inches, it makes a measurement. And it can do that at one spot in just a couple of minutes. We went to 34 sites around the whole lake and did this 3 times a week. So every day, we made 100,000 measurements, which is something that we couldn't do even 10 years ago. This wouldn't have been possible. So this instrument really helped us, saved us a lot of work, saved a lot of money too. And this is what some of that data looks like. This is a map of the lake. And each one of these circles is a station that we went to and dropped that instrument into the water column. And it's color-coded. So when you get down into the red and orange, like these guys, it tells us that there's not much nitrate there. The blue or purple guys here correspond to a lot of nitrate. And so this helped us to determine where we had to add more. So on August 31st, when we saw this map, we said, geez, no sense going here. There's plenty of nitrate there. But geez, it wouldn't be a bad idea to go over here and add some there. Otherwise, it would have been pure guesswork. We wouldn't know where on the lake that we needed it. And it's a big lake when you have to fill the whole thing with nitrate. So, we're getting toward the end of the scientific method here, so now we need to develop a conclusion. We need to decide, what are we going to tell people about this? Did it work? Didn't it work? Well, here's some more data for you. 2006, which is the blue circles here, is a year when we didn't add any nitrate to the lake. And in 2011, the yellow circles, we did add nitrate to the lake. And these are methyl mercury concentrations, again in nanograms per liter. 2006, they got really high, up around 10 or 15. In 2011, barely moves from zero. Very, very low concentrations. So, it worked. It definitely worked. It did what we thought it would do. Next question is, well, how much did that reduce upper water concentrations, where the fish live? Well, in 2006 and other years, we get this big spike in the fall that was associated with this mercury from the lower waters. In 2011, you can see no spike in the fall. And the reason is there wasn't anything in the lower waters to mix into the upper waters. So to us, this was great. We were pretty relieved. It looked like it worked. But no one will believe you if you only do it for 1 year, right? Maybe you just got lucky. So we did it again in 2012, and we did it again this past summer. So we did it for 3 years in a row. And in the other 2 years, the mercury numbers were even lower than this. But they're getting so low that you can barely see them anyway. So definitely worked. Our conclusion is that nitrate addition is a good way to keep mercury out of the water for the long term. So we had one more step to do, communicate the information, which we're doing some of that here today, right? But for scientists, what really counts, again, are these peer-reviewed publications. And we're proud to say that earlier this year, we wrote up an article, sent it out to reviewers to look at it, and they thought it was pretty good, so they published our article in their journal, which we're awful proud of. And so that's it. We covered the whole scientific method, and if you guys have any questions, I'd be glad to answer them or try anyway. Yes?
How much pounds or whatever you put nitrate you put in the water every time you went out?
On average, it was about 1 metric ton per day.
Okay.
If you average it over the whole summer. So a metric ton is, excuse me, 1,000 kilograms.
Okay.
And a kilogram is what, 2.2 pounds? Good. Yeah.
What was like your role in this? What did you do?
Well, I had, I helped develop the initial idea. It was me and Dr. Steve Effler, who I work with, and Charlie Driscoll, who's a professor at Syracuse University. So we had the original idea, and then we pitched that idea to Honeywell and to New York State DEC, and then I continue to work on the project overseeing it.
Because we read like an article on it. And yeah.
Yeah.
Do you know how much pollution
I'll let you next.
was put into the lake by the oil city and the oil tanks?
Boy, Oil City, I know that the groundwater in that area was contaminated. That, I can't tell you how much of that got into the lake. That I don't know. That really isn't related to this.
Climate change didn't affect any of the invasive species?
There are invasive species that we're interested in, but I'm not directly involved in trying to eliminate any of those. One I can talk to you about, have you ever heard of alewife? Yeah, there's a lot of those in the lake now, and they eat all the zooplankton. And the reason that's bad is because the zooplankton eat the algae. So it kind of works out that the more alewife you have, the more algae you have. Because they eat the guys that eat the algae. And that means that the lake isn't as clear as it could be. And there are years when the alewives aren't around and the lake gets really clear. It's really real nice to look at. And it ends up that we're like at the northern extent of their range. So in winters where the water gets really cold, all the alewife die off. And we end up with clear water the next year. In years where the lake stays warm, like it does a little south of here, we have alewives and we have muddier water. So, yeah.
Were you putting the nitrate in like the shallower waters where they were dredging and tapping the sun?
Nope, we only stayed, in the deep waters. Yeah, generally fit down 50, 60 feet. Yeah, pretty deep. Yes?
How much does this nitrate, like this, when you put it in, the ton you put in there, how much does it cost and who pays for it?
This is a Honeywell-funded project. So they pay for the nitrate and for all the labor to put it in the barge. Kind of soup to nuts. I don't know what it costs. I don't deal with that part of it. A lot, I'm sure.
What happens when the nitrate ends in the lake when they inject it into the water run out?
That's a good question. I didn't address that at all. There's something called a frac